How do you read a history book that was written over billions of years? A book written in a language that's completely invisible to the naked eye.
And maybe more importantly, how do you then pick up a pen and start editing that story yourself?
That's really the core of it, isn't it? That's the transition we're talking about today. It's this incredible shift from being, well, passive observers of biology
Right, like taxonomists or anatomists just cataloging what's there.
to becoming active molecular engineers. Welcome back to the Deep Dive. Today, we're jumping into a huge topic: DNA-based information technologies.
This is the toolkit. This is how we access, copy, read, and now even rewrite the genome.
And for this discussion, we are weaving together two essential texts. We're using the deep conceptual framework from Leninger's Principles of Biochemistry.
And grounding that with the clinical applications you find in Harper's Illustrated Biochemistry.
Now, we have to say this right at the top. This conversation, this deep dive,
Uh-huh.
it's a conceptual map. It's a synthesis of these ideas.
Exactly. We want to walk you through the logic, the why behind these technologies, but it is absolutely not a substitute for sitting down with the books themselves.
You've got to do the reading.
You really do. Leninger gives you those beautiful, detailed mechanisms, and Harper shows you why a doctor needs to know them. Our goal here is to build a kind of mental scaffolding for you.
So that when you see a complex diagram of, say, a cloning vector or a PCR cycle, it clicks. It makes intuitive sense.
It's the tour guide before you walk into the museum. You still need to look at the exhibits yourself.
And the main exhibit today is this idea the texts call the malleable archive. I love that phrase.
It's so powerful because it captures the whole paradigm shift. For most of scientific history, the genome was seen as this static, unchangeable book of life.
Locked away in a vault.
Totally. It was in the nucleus, it was protected, you inherited it, and that was that. Game over.
But the reality is so different.
Completely. We now know the genome is a working document. It's dynamic. And with these technologies, we've learned it's malleable. That's a key term from Leninger. We can get in there.
We can interrogate it.
We can cut it, we can copy it, we can even rewrite it.
So before we get into the really cool stuff, the CRISPRs and the sequencers, Leninger lays out what he calls 3 core truths about all of this. I found this to be a really helpful way to organize everything.
It's a great framework. The first truth is that the genome is the ultimate source.
Okay.
It contains the complete set of instructions, the haploid genetic complement for everything, the master blueprint for building and running an organism.
From eyelashes to enzymes.
Everything. But, having a blueprint locked in a safe doesn't do you much good.
Right, which brings us to the second truth: accessibility.
We can now read the code. We've moved past just inferring genetics by watching pea plants. We can directly sequence the A's, T's, C's, and G's. We can see the letters.
And the third truth, this is the one that really defines our era.
Malleability. This is it. This is the big shift. We can now change the code to alter how an organism works.
We can move a gene from a jellyfish into a bacterium to make it glow.
Or, more importantly, we can fix a disease-causing mutation in a human cell. We are not just reading the book anymore. We are authors.
Okay, but there's a huge fundamental problem standing in the way of all this. It's a problem of scale.
It is the ultimate needle in a haystack problem.
Because a chromosome is it's enormous. It's millions or even billions of base pairs long.
And the gene you're interested in, the gene that causes cystic fibrosis, for example, might only be a few thousand base pairs.
So you're looking for a tiny specific sentence in a library full of encyclopedias.
And it's worse than that because the needle looks chemically identical to the hay. The sugar phosphate backbone is the same everywhere. It's all DNA.
So how do you find that one specific segment, pull it out, and then make enough copies of it to actually study?
That is the mission. And this deep dive is about building the toolkit that solves that very problem. Let's open the toolbox.
All right, section 1. This is where it all started. Recombinant DNA basics. I think of this as the cut and paste era of molecular biology.
That's a perfect description. This is the foundation for everything that followed, and it's all about making clones.
Okay, let's pause there, because when you say clone, people immediately think of Dolly the sheep or something from a sci-fi movie.
Right, but that's not what we mean here. In this context, a clone is just a population of identical molecules or identical cells that all came from one common ancestor.
So we're not cloning a whole organism. We're cloning a single piece of DNA.
Exactly. We're making millions and millions of identical copies of one page from that giant encyclopedia so that we have enough to read it and work with it.
Leninger gives us a really logical 5-step process for cloning. I think it's worth walking through because it's the fundamental workflow.
It is. Step 1, you have to obtain the DNA segment you care about. You have to find your needle.
Step 2?
You need a carrier, a vector. You can't just toss a naked piece of DNA into a cell. The cell's defenses will chew it up. You need a delivery vehicle.
Okay, so you have your DNA and your vehicle. Step 3.
Ligation. You have to join them together. You need to glue your gene of interest into the vector.
And then step 4.
Transformation. That's the act of moving that newly combined molecule, the recombinant DNA, into a host organism. Usually it's something simple like an E. coli bacterium.
And the final step, step 5.
Selection. Because the transformation process is really inefficient, most of the bacteria won't take up your new DNA, so you have to find the few that did. You have to select for the winners.
Okay, that's the roadmap. Now let's dig into the tools that make each step possible. So, step 1, obtaining the DNA. We need a way to cut it out of the chromosome. We need molecular scissors.
And for that, we turn to the restriction endonucleases.
I love the story behind these. Werner Arber didn't just invent them, he discovered them in nature.
It's a beautiful example of how basic, curiosity-driven science leads to a revolution. He was studying how bacteria defend themselves against viruses, against bacteriophages.
And he found they have an immune system.
A primitive but effective one. They produce these enzymes that restrict or cleave any foreign DNA that gets injected by a virus. It chops up the invader's genome before it can take over.
So nature had already invented the perfect tool.
And scientists figured out how to harness it. Specifically, we use a class called type 2 restriction enzymes.
Why type 2? What's special about them?
Precision and simplicity. Other types, like type I, are a bit messy. They recognize a specific sequence but then cut the DNA at some random location far away.
Not very useful if you need a precise cut.
Not at all. Type 2 enzymes are perfect. They recognize a very specific sequence of DNA, usually 4 to 6 base pairs long, and they cut right there, inside that sequence, every single time. And they don't need any AT key to do it. They're simple, reliable machines.
Let's talk about those sequences they recognize. They have this interesting property. They're palindromes.
Yes. So in English, a palindrome is a word like level or race car, reads the same forwards and backwards. In DNA, a palindromic sequence is one where the top strand read 5' to 3'.
Is the same as the bottom strand, also read 5' to 3'.
Exactly. A famous example is the recognition site for the enzyme E. coli. It's GAATT on the top strand.
Which means the bottom strand is CTT egg. So if you read the bottom strand from right to left in its 5 to 3 direction, it's also GATTC.
Perfect. Now, the way the enzyme cuts that sequence is incredibly important. This is the difference between blunt ends and sticky ends.
Okay.
A blunt end cutter would slice straight through both strands, like chopping a carrot. Clean break. A sticky end cutter, like E. coli, makes a staggered cut.
What does that mean, staggered?
E. coli cuts between the G and the A on both strands. Because the strands are anti-parallel, this doesn't happen in the same place. It leaves a little single-stranded overhang on each piece.
A few unpaired bases just hanging out there.
Yes. In this case, the sequence ATT is left hanging off, and those are the sticky ends.
And they're sticky because A wants to pair with T. They're chemically attracted to each other.
They desperately want to form hydrogen bonds. This is the genius of it. If you cut your human gene with E. coli, and you cut your bacterial vector with E. coli,
they will both have the same sticky ends, AATT.
And they will be perfectly complementary. They're like little pieces of Velcro. When you mix them together in a test tube, the sticky end from the human DNA will find and anneal to the sticky end of the vector. It holds them together in the perfect orientation.
But those hydrogen bonds, they're not permanent, right? They're relatively weak.
Correct. They hold the pieces in alignment, but the sugar phosphate backbone is still broken. That's where you need tool number 2.
DNA ligase.
The molecular glue. Or a welder, maybe. It comes in and forms a permanent covalent bond, a phosphodiester bond, to seal that gap in the backbone. It makes the 2 separate pieces of DNA into one continuous, stable molecule.
And just like that, you've created what Harper calls a chimeric molecule, recombinant DNA.
A molecule that has never existed in nature before. A piece of human DNA physically joined to a piece of bacterial DNA.
Okay, let's talk more about that bacterial part, the carrier, the vector. You mentioned it's usually a plasmid.
Right. Plasmids are these little extra circles of DNA that bacteria have. They're separate from the main bacterial chromosome. Think of them like little USB drives that bacteria can swap between themselves.
Trading useful files, like antibiotic resistance genes.
Exactly. Hey, here's a plasmid that will help you survive penicillin. We've hijacked that system. But the plasmids we use in the lab aren't just natural plasmids. They've been engineered with 3 key features.
Okay, feature 1.
An origin of replication, or ORI. This is the start here signal for the bacterium's own DNA copying machinery. Without an ORI, your plasmid would just sit there. When the bacterium divides, it wouldn't be copied, and it would be lost within a few generations.
So the ORI ensures that every time the bacterium copies its own chromosome, it also makes copies of your plasmid.
Exactly. It makes sure your factory keeps running.
Feature 2.
The selectable marker. This is the key to solving that selection problem we mentioned. Getting the plasmid into the bacteria is called transformation, and it's incredibly inefficient. Maybe only 1 in 10,000 cells actually takes it up.
So you have a tube of 10,000 bacteria, and only one has your precious recombinant DNA. How do you find it?
You create a life or death test. The plasmid carries a gene that confers resistance to an antibiotic, like ampicillin. So you take all your bacteria and you spread them on a petri dish that's full of ampicillin.
It's a hostile environment.
For most of them, yes. The 9,999 bacteria that failed to take up the plasmid have no defense against the ampicillin. They die.
But the one that succeeded.
The one that has the plasmid also has the resistance gene. It survives, and it divides and divides and forms a visible colony. So any colony you see growing on that plate is, by definition, a clone derived from a bacterium that successfully took up your plasmid.
That's a brilliant bit of logic.
Yeah.
And the third feature?
That's the multiple cloning site, or MCS. This is a short stretch of DNA that we've engineered into the plasmid to be packed with recognition sites for dozens of different restriction enzymes.
It's the cargo bay.
It's the universal adapter. It gives you the flexibility to use whatever scissors, EcoRI, BAM-Hi, Hin3A work best for cutting out your gene of interest. You have lots of options for where to paste your cargo.
So this whole process, cutting, pasting, transforming, selecting, and growing bacteria. It's powerful, but I imagine it takes time.
It does. Growing up bacterial cultures takes at least a day, often longer. It's the slow part of the process.
And in a clinical setting, for diagnostics, you don't always have days. You need answers in hours.
Which brings us to the next great revolution. The technology that took DNA amplification out of the cell and put it into a test tube.
Section 2, the photocopier.
Otherwise known as the polymerase chain reaction, or PCR.
This might be the most famous technique in all of molecular biology. Harper makes it clear that this changed everything. Forensics, diagnostics, evolutionary biology.
It really did. Before PCR, if you had a tiny speck of blood at a crime scene or a single hair follicle, there just wasn't enough DNA to analyze. You needed a relatively large sample.
PCR changed the math on that.
Completely. It gives us the ability to find that one specific needle in the haystack and make billions of copies of just the needle, ignoring all the hay. You can go from a single molecule of DNA to an amount you can see on a gel in just a few hours.
And the mechanism is beautiful in its simplicity. It's all based on cycling through different temperatures.
That's all it is. 3 steps in a repeating cycle. Step 1 is denaturation.
This is the hot step.
Very hot. Around 95 degrees Celsius. This is enough heat to break the hydrogen bonds holding the 2 strands of the DNA double helix together. You are literally melting the DNA apart into single strands.
Step 2 is annealing.
For this, you cool the reaction down, usually to somewhere between 50 and 65 degrees Celsius. In this cooler environment, things can stick together again. And what we want to stick are our primers.
The primers are the key to specificity, right? This is how you tell the reaction what to copy.
Absolutely. The primers are short, single-stranded pieces of DNA, maybe 20 nucleotides long, that we synthesize in the lab. We design them to be perfectly complementary to the sequences that flank our gene of interest. They are the bookends.
So out of the whole genome that's in the tube, these little primers will only bind to one specific spot.
That's the theory. They find their target and they anneal, providing a starting point for the next step.
Step 3, extension.
Here, you raise the temperature again, this time to about 72 degrees Celsius. This is the ideal working temperature for our special polymerase enzyme. The enzyme latches onto the primer and starts adding nucleotides, extending the strand and making a new copy of the template DNA.
So at the end of one cycle, you've gone from 1 copy of your target sequence to 2.
Correct. And then you just repeat it. Heat to 95 to separate everything, cool to 55 to let more primers bind, heat to 72 to extend again.
And with each cycle, the number of copies doubles.
It's an exponential explosion. After just 20 cycles, you have about 1 million copies. After 30 cycles, you have over 1 billion. It's an incredibly powerful amplification engine.
You mentioned a special polymerase. This is the story of Taq polymerase, right? Why couldn't the pioneers of PCR just use regular human DNA polymerase?
Well, think about that first step. Denaturation at 95 degrees Celsius, that's almost boiling. What happens if you put a human protein in boiling water?
It denatures. It unfolds. It's cooked.
It's useless. In the very early days, the inventor, Carey Mullis, had to physically open the tube after every single heating cycle and add a fresh drop of enzyme. It was incredibly tedious.
So the search was on for a heat-proof enzyme.
Exactly. And the search led to the hot springs of Yellowstone National Park, where there are bacteria that thrive in near boiling water. One of them is called Thermus aquaticus.
Ah, so Taq polymerase is from Thermus aquaticus.
Precisely. Its enzymes, including its DNA polymerase, are naturally stable at very high temperatures. Taq polymerase can go through that 95-degree step over and over again without breaking a sweat.
And that single discovery is what allowed the whole process to be automated.
It made it possible to build a machine, put all your reagents in a tube, press start, and come back 3 hours later to 1 billion copies of your DNA. It democratized the whole field.
Now, Harper's talks about a couple of important variations on PCR that are critical in medicine. We hear the acronyms all the time, RT-PCR and qPCR.
Let's break those down. RT-PCR stands for reverse transcriptase PCR. This is what you use when your starting material isn't DNA, but RNA.
And why would you want to start with RNA?
A couple key reasons. First, many viruses have RNA genomes. Think of HIV, influenza, or the SARS-CoV-2 virus that causes COVID. To detect them with PCR, you first have to convert their RNA into a more stable DNA copy.
And the enzyme that reads RNA and writes DNA is reverse transcriptase.
Exactly. It creates a DNA copy, which we call complementary DNA, or cDNA. Then you can run a standard PCR on that cDNA to amplify it.
What's the other reason?
To study gene expression. Your genome is the same in almost every cell, but a liver cell expresses a different set of genes than a neuron does. If you want to know if a specific gene is turned on in a cell, you look for its messenger RNA, its mRNA.
So by doing RT-PCR on the mRNA from a tissue sample, you can see which genes are actively being transcribed.
Precisely. It tells you what the cell is doing, not just what it's capable of doing.
Okay, so that's RT-PCR. What about qPCR? The Q is for quantitative.
Yes. Standard PCR is a yes or no answer. At the end of 30 cycles, is there a product, yes or no? qPCR, or quantitative PCR, tells you how much of your target DNA was there to begin with.
How does it measure that?
It's clever. You add a fluorescent probe to the reaction that's designed to glow only when it binds to the newly made PCR product. A camera inside the machine measures the intensity of that glow in real time after every single cycle.
So if you start with a lot of DNA,
the glow will get bright very quickly. It'll cross a certain threshold of brightness in, say, only 15 cycles.
And this is where that term seat value comes from, which we all heard about during the pandemic.
The cycle threshold. A low seat value, like 15, means you started with a ton of viral RNA. The patient has a high viral load. If it takes 35 or 40 cycles for the fluorescence to become detectable, that's a high seat value. It means you started with very little virus.
So it's not just a diagnosis, it's a measure of the severity of the infection.
Exactly. It's a powerful clinical tool.
Okay, so we can cut, paste, and copy DNA. But all of that is kind of useless if we can't actually read what it says.
Which brings us to Section 3, reading the manuscript, the world of DNA sequencing.
The texts draw a clear line between the old way, Sanger sequencing, and the modern next-generation methods. Let's start with the classic, Sanger sequencing.
This was the workhorse for decades. It's an incredibly elegant method based on a principle called chain termination.
I like the analogy of a construction crew building a brick wall.
That's a great one. So imagine DNA polymerase is your brick layer. The normal bricks are the 4 standard nucleotides, the DNTPs. And to add a new brick, the brick layer needs a specific spot on the previous brick to connect to. That's the 3' hydroxyl group.
So a normal brick has a connector.
Always. But what Fred Sanger did was create a few defective bricks. These are called deoxynucleotides, or DDNTPs. They look almost identical to the normal bricks, but they're missing that 3' hydroxyl connector.
So if the bricklayer accidentally grabs one of these defective bricks and puts it on the wall,
the wall stops. He can't add another brick on top of it. The chain is terminated.
So in the lab, you set up a reaction with mostly normal bricks, but you sprinkle in a small amount of these defective chain-terminating bricks for each of the 4 letters.
Exactly. And each type of defective brick, the A, the T, the C, and the G, is labeled with a different colored fluorescent dye. So as the polymerase is copying the DNA, it will randomly incorporate one of these dideoxynucleotides, and the chain will stop. This happens at every possible position. What you're left with is a collection of DNA fragments of every possible length, each ending with a specific colored letter.
And then you sort them by size.
Right. You run them through a very thin capillary tube filled with a gel. The smallest fragments move the fastest. A laser at the end of the tube reads the color of each fragment as it passes by.
So if the first fragment to come out is green, you know the first letter is an A. If the next is blue, the next letter is a C. You're literally reading the sequence of colors.
That's it. It's beautiful. And that is exactly how the first Human Genome Project was done.
But it was slow and incredibly expensive.
Monumentally so. Over a decade and nearly $3 billion.
And today, I can get a report on my ancestry DNA for under $100.
That's the power of next-generation sequencing, or NGS. The texts also call it high-throughput sequencing.
What was the fundamental shift? What changed?
Massive parallelization. Sanger sequencing was like one bricklayer building one wall very carefully. NGS is like having millions of bricklayers on a single glass slide, all building their own little walls simultaneously.
It's factory-level production.
Millions of sequencing reactions happening at once, and a high-speed camera takes pictures after each nucleotide is added, recording the flash of colored light at each spot. We generate unbelievable amounts of data terabytes from a single run.
And Harper's points to a really important clinical application of this, which is sequencing the exome.
This is all about being efficient. The full human genome is over 3 billion base pairs, but the parts that actually code for proteins make up only about 1.5% of that. That 1.5% is the exome.
And so the rest of it is regulatory sequences, or what used to be called junk DNA.
Right. And if you're a clinician looking for the cause of a rare genetic disease, the mutation is almost certainly going to be in the exome, in one of those protein-coding genes.
So why read the entire encyclopedia when you can just read the chapters that matter for your question?
Precisely. Sequencing just the exome is much faster and cheaper. You can get it done for a few $100s now. It's becoming a standard diagnostic tool.
But the technology doesn't stop there.
Yeah.
The outline mentions a third generation of sequencing, PacBio and Nanopore. This stuff sounds like it's from the future.
They solve a specific problem that NGS has. NGS gives you what we call short reads. It's like taking a book, shredding it into millions of tiny 150-letter-long snippets, and then trying to piece the book back together.
That sounds hard, especially if there are repetitive parts.
That's the exact problem. If a paragraph is just the blue sky, over and over, how do you know how many times it repeated? The short reads can't tell you.
So third gen gives you long reads.
Very long reads. Nanopore technology is just wild. There are no lasers, no cameras. It's all electrical.
How did that work?
You take a membrane with a tiny nanometer-sized hole in it, a nanopore. You pass an ionic current through that pore. Then you take a single long molecule of DNA and you thread it through the pore. As the DNA molecule passes through, each base, A, C, T, and G, has a slightly different shape and chemical property. Each one disrupts the flow of that electrical current in a unique, measurable way.
You're reading the sequence by measuring changes in electricity.
In real time on a device the size of a USB stick.
Yeah.
And because you're reading a single unbroken molecule, you can get reads that are tens of thousands or even hundreds of thousands of bases long. It lets us finally assemble those really complex, repetitive parts of the genome.
That's amazing. Okay, let's shift gears a bit to section 4, the librarian. We have all this information, but how do we organize it and search it? Let's talk about libraries and blotting.
Right. We've used the term library before, but it's important to distinguish between two main types, genomic libraries and cDNA libraries.
A genomic library, I'm guessing, contains everything.
The whole shebang. Every piece of DNA from the organism's chromosomes, that means all the genes, but also all the introns inside the genes, all the regulatory sequences, all the junk DNA between genes. It's the complete, unabridged blueprint.
And when would you want that?
If you're studying the fundamental architecture of a genome, or looking for a regulatory element like a promoter.
But what if my goal is more practical? Let's say I want to engineer a bacterium to produce human insulin for diabetic patients.
Ah, now you've hit on the problem. Bacterial cells are simple. They don't have the machinery to splice out human introns. If you put the entire human genomic insulin gene into an E. coli, it will try to translate the introns along with the exons.
And you'll get a useless, garbled protein.
Exactly. For that, you need a cDNA library. Remember, cDNA is made from messenger RNA using reverse transcriptase, and the cell has already done the hard work of splicing out the introns to create that mRNA.
So a cDNA library only contains the protein coding sequences. the exons?
It's the collection of only the genes that were actively being expressed in the tissue you took the mRNA from. It's the edited, ready-to-translate version that bacteria can understand. This is why virtually all recombinant protein drugs are made using cDNA.
Okay, so you have your library, but you still need to find one specific book on the shelf. That brings us to blotting. I can never keep the compass directions straight for these.
It's a classic bit of scientific humor. It all started in the 1970s with a biologist named Ed Southern. He developed a brilliant technique to transfer DNA fragments from a gel to a membrane so you could search for a specific sequence.
And they named it the Southern blot after him.
Correct. Southern equals DNA. Then, a few years later, other scientists developed a similar method for detecting RNA. And as a joke,
They called it a Northern blot.
naturally. And then when the technique was adapted for protein,
It had to be a Western blot.
exactly. And the Western blot is the one Harper really emphasizes because it's still a cornerstone of clinical diagnostics.
What's a classic example?
Confirmatory HIV testing. The first test you get is usually a rapid screening test called an ELISA. It's very sensitive, but it can sometimes give false positives.
So if that comes back positive, you need a more specific follow-up test.
That's the Western blot. You take the patient's blood serum, which contains their antibodies, and you test whether those antibodies stick to specific HIV proteins that have been separated by size on a membrane.
So it doesn't just say HIV positive. It says, yes, I see an antibody response to the P24 capsid protein and to the GP120 envelope protein. It's definitive proof.
It's highly specific. It confirms the diagnosis. So Southern for DNA, Northern for RNA, Western for protein, the librarian's search tools.
Which brings us to the technology that's really dominated the last decade. Section 5, the editor.
CRISPR-Cas9.
We started by saying the genome is malleable. But for a long time, the tools for actually editing it in the living cell were
Well, they were clumsy.
incredibly clumsy. It was like trying to do brain surgery with a sledgehammer. The tools we had were difficult to design, expensive, and not very efficient. CRISPR changed all of that because it's programmable.
And once again, this amazing tool didn't come from a biotech lab initially. It came from bacteria. It's the same story as restriction enzymes. It's another bacterial immune system. But this one has a memory. When a bacterium is attacked by a virus and survives, it actually cuts out a small piece of the viral DNA and stores it in its own genome in a special region called the CRISPR array.
It keeps a molecular mugshot of its enemies.
It's a genetic most wanted list. Then, if that same virus ever tries to infect the cell again, the bacterium can quickly make an RNA copy of that mugshot to guide an enzyme to find and destroy the invader's DNA.
So let's break down the system we use in the lab. There are two key components, Cas9 and the sgRNA.
Right. Cas9 is a protein, specifically a nuclease. Think of it as the molecular scissors or the blade. Its only job is to cut DNA. But the thing is, Cas9 is blind. It has no idea where to cut.
It needs a guide dog.
A very specific guide dog. That's the sgRNA, which stands for single guide RNA. This is a synthetic piece of RNA that has two parts. One part acts as a handle that grabs onto the Cas9 protein. The other part is a sequence of about 20 nucleotides that we design in the lab to be a perfect match for the target DNA sequence we want to cut.
So if we want to target the mutation that causes sickle cell anemia, we design those 20 letters to match the sequence of the broken hemoglobin gene.
Exactly. The sgRNA and the Cas9 protein form a complex. The RNA then acts as a homing beacon, scanning the 3 billion letters of the genome until it finds its one perfect match.
And when it finds it, it docks, and Cas9 makes the cut.
A clean double-strand break right at that precise location.
Now, this is a really important point. The cut itself is not the edit.
No, the cut is just the damage. The magic, the editing, happens when the cell tries to repair that damage.
And the cell has two different ways of doing that, which we can hijack for our own purposes.
Right. The first and most common repair pathway is called NHEJ, non-homologous end joining.
This sounds messy.
It's the cell's panicked emergency response. It basically just grabs the two broken ends of the DNA and shoves them back together. It's fast, but it's sloppy. In the process, it usually deletes a few bases or inserts a few random ones.
It creates a small mutation, a scar, at the cut site.
Exactly. And that small scar is often enough to disrupt the reading frame of the gene, effectively breaking it. So if your goal is just to turn a gene off, what we call a knockout, NHJ is perfect.
But to cure sickle cell, we don't want to break the gene. We want to correct the typo.
For that, you need the second pathway, HDR, or homology-directed repair.
This is the more precise method.
Much more precise. The cell's natural HDR pathway looks for a template, usually the sister chromosome, to use as a guide for a perfect repair. In the lab, we can trick it by providing our own template.
So along with the CRISPR-Cas9 system, we also flood the cell with a short piece of DNA that contains the correct sequence for the hemoglobin gene.
Exactly. The cell sees the double-strand break, it sees our template, and it uses that template to patch the break. In the process, it writes our correction directly into the patient's genome. It's like a find and replace function for DNA. This is a knock-in.
And Harper's highlights these single gene disorders like sickle cell or thalassemia as the perfect first targets for these therapies.
And they are. Clinical trials are showing incredible results. We are seeing functional cures, but we have to talk about the risks.
There's always a but.
The biggest technical concern is off-target effects.
The guide RNA is only 20 letters long. In a genome of 3 billion letters, what are the chances that a similar-looking sequence exists somewhere else by accident?
The chances are not zero. You might have a perfect match at your target on chromosome 11, but a near-perfect match may be 19 out of 20 letters on chromosome 4.
And the Cas9 might go there and make a cut by mistake.
And if that accidental cut happens to land in the middle of a vital tumor suppressor gene, you could potentially cure one disease while causing cancer. A tremendous amount of research goes into designing guides that are ultra-specific to minimize this risk.
And then there's the big ethical line, the difference between editing somatic cells and germline cells.
This is the crucial distinction. Somatic cells are all the non-reproductive cells in your body, your blood cells, skin cells, liver cells. If we take a patient's bone marrow stem cells, edit them to fix the sickle cell gene, and put them back in, those changes stay with that patient.
The edits die when the patient dies. They are not inherited.
Correct. But if you edit the germline sperm, eggs, or a very early embryo, those changes become a permanent part of that individual's genetic makeup. They will be passed down to their children and their children's children forever.
You are permanently altering the human gene pool.
You are engaging in heritable human genome editing, and that is a line that carries an enormous weight of ethical and societal responsibility.
Let's zoom out a little for our last section, section 6, the context. Because genes don't act alone. It's about how they fit into a larger system.
We're moving from a one-gene-at-a-time perspective to a true systems biology approach.
But before you can study the system, you still have to find the disease genes in the first place. How did we find the genes for something complex like Alzheimer's disease?
Through old-fashioned detective work called linkage analysis.
This involves looking at big family trees, right?
Yeah.
Pedigrees.
Exactly. Leninger uses the example of early-onset Alzheimer's. Researchers studied large families where this devastating disease was passed down from one generation to the next. They sequenced bits of DNA from everyone, both sick and healthy family members.
And they looked for genetic markers that were different between the two groups.
They looked for markers, little variants called SNP, single nucleotide polymorphisms, that were always inherited along with the disease. They found a specific set of markers on chromosome 14 that tracked perfectly with who got sick.
It's genetic guilt by association.
Precisely. It told them the gene had to be in that neighborhood on chromosome 14. That narrowed the search from 3 billion base pairs down to a few million, which eventually led them to identify the PS1 gene.
So linkage tells you where the gene is. But what about how it's being controlled? This brings us to a technique with a very intimidating name. ChIP.
Chromatin immunoprecipitation. It sounds terrible, but the idea is actually really cool. It answers the question, which proteins are physically touching which parts of the DNA right now?
So you can see if a specific transcription factor protein is sitting on the promoter of the insulin gene, for example.
Exactly. You use a chemical to freeze everything in place, linking the proteins to the DNA they're bound to. Then you use an antibody to grab your protein of interest and you pull out the DNA that was stuck to it. Then you just sequence that little piece of DNA.
It's like finding a book in the library and seeing whose fingerprints are on it.
That's a great way to think about it. It helps us build the regulatory wiring diagram of the cell.
And when you combine all of this, genomics, transcriptomics, RNA, proteomics, proteins, metabolomics, metabolites, you get to this idea of systems biology.
It's the understanding that a complex disease like type 2 diabetes isn't just one broken gene, it's a failure in the network. A subtle change in dozens of genes interacting with diet and lifestyle leads to the collapse of the system.
To treat the disease, you have to understand the system.
And that's the future of medicine.
Before we wrap up, there's one last story from the notes that I just find fascinating. It's about using these tools for time travel, in a way. Ancient DNA.
This is one of the most incredible applications of NGS technology. We can now pull tiny fragments of DNA out of bones and teeth that are tens of thousands or even hundreds of thousands of years old.
But isn't that DNA a mess? Degraded and covered in bacterial DNA from the soil?
It's a total mess. It's shattered into tiny pieces, and it's maybe 99% contamination. But because NGS is so powerful, we can sequence everything, the human bits and the bacterial bits, and then use computers to filter out the contamination and stitch the ancient genome back together.
And in doing so, we learned something profound about ourselves.
We sequenced the Neanderthal genome and the Denisovan genome, and we found that for most people outside of Africa, these aren't just extinct cousins, they're part of our ancestry.
Our ancestors interbred with them.
They did. And we still carry their DNA in our cells today. Some of those ancient genes are involved in our immune system and helped our ancestors adapt to new environments as they spread across the globe.
It's incredible. So we've covered a huge amount of ground today. We started with the concept of the malleable archive. We picked up our molecular scissors and glue.
The restriction enzymes and ligase.
We loaded them onto delivery trucks, the vectors. We fired up the photocopier's PCR. We learned how to read the manuscript with sequencing.
And finally, we picked up the editor's pen with CRISPR. It's a complete toolkit for interrogating and engineering the code of life.
So what's the big so what for the listener? How does all this change the world for a medical student or just anyone who's curious?
The big takeaway is that diagnosis and treatment are fundamentally shifting. We're moving from looking at symptoms and organs to looking at molecules and sequences.
It's no longer one-size-fits-all medicine.
Not at all. It's becoming personalized medicine. We can tailor treatments to a patient's specific genetic makeup or to the specific mutations driving their particular cancer.
It's an incredibly empowering moment in history.
It is. But with that power comes a profound responsibility.
How so?
Because we have made the full transition from being simply the readers of our biological history to becoming the potential authors of its future.
The question is no longer can we do it.
The question is, should we? And more pointedly, do we understand the staggering complexity of these biological systems well enough to predict the long-term consequences of the edits we make?
That is the thought to end on. The archive is open, the pen is in our hand, and now we have to decide what to write.
Indeed.
Thank you for taking this deep dive with us today.
It was my pleasure.
Now, seriously, go open Leninger, go open Harper, find these chapters, look at the diagrams of the cloning sites, the PCR cycles, the Cas9 protein complex. They're beautiful little machines, and now that you know the story, they're going to make a whole lot more sense. We'll see you on the next Deep Dive.